Anybody there? Oh, it's Michael again. Michael, I don't know what's going on. I click to log on and I get switched to my own Zoom channel rather than the department's.
Okay, can you hear me now?
I can hear, I think is that Michael or Chris?
I'm Michael.
Michael, I don't know what's going on. I click on the link that I sent to everybody, and it brings up my personal meeting, the one I used to teach, rather than the department's Zoom. So I'm really a little bit puzzled as to what the hell is going on.
I don't know, Kevin, but I think we're all on the right Zoom now. Oh.
Is that Rich Rand?
Yeah, Chris is with me.
Rich, I'm really confused. I clicked on the link that I sent out, which is the department link, and yet it brought up my own personal Zoom link. And yet you're on it, and Michael Holzheider is on it, or was a moment ago.
Still am, yeah.
Yeah, no, I don't know what happened, but I think it's all okay as far as everybody's concerned.
Okay.
And here's our speaker.
Hey.
Oh, wonderful. Hello, Professor Yeomans.
Hello, hi, I'm Julia.
How do you pronounce your name?
Yeomans is right, yes.
Yeomans?
Yeomans, yes,
Okay.
yes.
Sorry about typing Julie or having Julie instead of Julia in the
Oh, it's not a problem. Not a problem at all. I just thought I didn't want it to go too far and proliferated too much.
Yeah, well, it was corrected in the second announcement.
Yeah, absolutely.
And I knew your name was Julia. It was just, I don't know what happened, actually. We seem to be having a bit of a problem here with Zoom at our end, but it may just be that people are late at logging on. The reason I'm confused is, well, that people in the department are a little confused, and I click on the link that I sent out, and it brings up Zoom with my personal Zoom link on it, something ending in 161, whereas the department one ends in 696. And in fact, the two numbers are totally different.
Oh, okay. Well, this one is 696, and that's the one I clicked.
And so you're on 696. Okay. That's the one I clicked on, but my computer thinks it's on 161. But I guess it'll be all right. Anyway, we all look forward to your talk, and I think it's really interesting that you and I guess other people are actually thinking about life in really physical terms, as opposed to just on the microscopic terms of chemical reactions, which I guess is what one does in biophysics and molecular biology. There, one talks about the reactions, but one doesn't think of them in terms of life because they just look like chemical reactions.
Yeah, it's a different way of thinking about it, but I'm sure it will take both. can I just try sharing my screen? Shall I just do that?
Sure, good
Um,
idea.
Oh, it says host disabled participant screen sharing. So you'll have to give me permission to do that.
Well, I'm going to hit screen share and I'll make it desktop 2, which, oh God, that's my desktop 2, not yours. That's even worse. Let's see, maybe I'm going to stop. I better stop share. Let me call the people there. So you have a problem there with sharing.
Yeah, I just need.
I mean, I have always said that the right way to do it is to have the host run the thing. Instead, we have somebody in the department running the thing, and he's not sure what he's doing. And I'm going to try to call
Okay.
him.
Yeah, you have to ask him to give me permission to share.
Yeah, hi. We have to give Professor Yeoman permission to share the screen. Well, if you click on that thing that says, let's see, where does it
If I click on share screen, it says host disabled participant screen sharing.
all right, I'm going to hit multiple participants can share simultaneously. Try now.
Yep, that works.
Okay, I managed to solve that problem apparently.
Yeah. Share.
Oh, there we are. Yes. Okay, we certainly see your screen. Kinesin walking from Inner Life of Cell. Great.
Yes.
Okay,
no.
Chris, I'll let you go. Thanks. Bye.
Show. I can start. Is that now it's full screen? Is that
It looks yes, it looks perfect. And it it reminds me of one of the BBC TV shows, which show a view very much like that. I'm gonna see if I can move this to this screen. Okay, that's better. And then I'm going to move these over there. Okay. Yeah, I do remember, though, being told once that what's actually going on in a cell is there are something like at least 500 chemical reactions occurring simultaneously, and they keep each other going, and that's why you have a sort of equilibrium or a stasis of some kind. I mean, not equilibrium in the sense that you were describing it, meaning death, but I guess a metastable state above equilibrium. If you have these 500 reactions in a simple bacterial cell, but presumably there are even more than 500. I guess he was or she was saying 500 principal reactions that happened all the time, and then there were special ones that turned on now and then. Anyway, I shouldn't be talking that much. We should be listening to you.
Yeah, we've got a while there till whatever time it is there.
I've known about Schrodinger's book for some time, but I never got around to reading it. Is it something you recommend reading?
The bits, I mean, I've read bits of it. I haven't read it all. I think it's quite interesting. I mean, a lot of it is wrong, but it's still very clever.
Yeah.
I mean, only wrong in that we know better now. We think we know better now.
Yeah. Yeah, obviously there's been so much progress in biology.
I like telling I mean, it's useful for my to tell my quantum colleagues who don't really approve of biology that Schrödinger thought about it as well. Makes it a bit more respectable.
Yeah, well, I'd like to see more of a connection between, molecular biology and biophysics, and between the two of them and medicine. I guess at my age, I'm more concerned about advancing medicine than understanding life.
Yeah, it's hard. Science
When I was 20, I didn't care so much about medicine, but I'm considerably older now.
So what's the weather like in New Mexico at the moment? Is it nice and
Oh,
warm?
it's always boring in New Mexico. Clear blue sky, bright sun, mild temperatures. Occasionally we get a
I
breeze.
was just going to show you.
Oh, you're going to give us a view of the...
Make me jealous of the sunshine.
Yeah, well, it is. It's beautiful sunshine, beautiful weather. It's a lovely climate. You should move here.
I actually, I'm in California, which I've been in many times, is wonderful for a while, but I do miss the rain after a while. And
Yeah,
also, what do you talk about? I mean,
no, you're right. We rejoice when we get a day of rain, but when we have 2 days of rain, we get tired of it.
yes.
But yeah, you're right. Of course, Oxford is of course a very interesting place. What about consciousness? Have you thought much about that, or worried about, or made any progress? I mean, it seems so hard.
No, I mean, that's the neuroscientists really. I mean,
Yeah.
I'm very much doing physics, and rather than worrying about things like that.
Yeah. Well, let's...
on the other screen, but I'm not so I can.
Let's see, we have, I think, 2 minutes before we should start getting serious. My computer says I'm recording, or at least somebody's recording. I don't know, it's so confused today. It's being recorded somewhere, somehow.
Yeah, it told me to accept the recording when I logged in. So probably it is being recorded somewhere, somehow.
I'm looking at my iWatch, not my iWatch, my iPhone. Okay, it's 3:30. We're very lucky today to have Professor Julia Yeomans of the University of Oxford Zoom us a colloquium. It's very kind of her to do that, especially since it's 10:30 in England. She's head of theoretical physics at Oxford University, and she's going to talk to us today about dense active matter, which is, I think, will change somewhat how we think about life. So please take the floor, Professor Yeomans.
Okay, well, thank you very much for the invitation. It's lovely to talk to you. Please ask questions just interrupt me during the talk, because it is quite late here, and it'd be good way of keeping me awake. So if I look sleepy, please just sort of ask a question. I'm going to talk about active matter. And what active matter is, it's particle systems which take energy from their surroundings and then use it to do things. So cells will take energy from the surroundings, chemical energy in the form of ATP, and use it to move or to divide or to do work. Or little microswimmers will use the energy to move around. And on a larger length scale, all animals are continually taking energy from their surroundings and using it to be alive, really, to do things. And so this is thinking of living systems as being out of equilibrium. Living systems are meant to be out of thermodynamic equilibrium. And that's what active matter means. It's the study of things which are existing out of thermodynamic equilibrium. And what I'm going to particularly talk about today is dense active matter. What happens if you take loads of these particles and put them together? And here's some pictures of what happens. The first one is a dense suspension of microswimmers, E. coli, about 10 microns long. And you can see that when you put them together, what you end up with is this swirling motion, something which looks like turbulence. Here's another one. This is a layer of epithelial cells, which have been taken out of the body and put on a Petri dish. And again, they're moving the whole time, and they are moving in a random way. And again, you get a turbulent-like behavior. And the last one I'm going to show you is, again, this turbulence. It's called active turbulence. And what this is microtubules, long polymers, driven around by motor proteins. So immediately there are questions. These things are tiny, and tiny means low Reynolds number. And normally at low Reynolds number, flows are very gentle and smooth. And here we're seeing something which looks like turbulence. So we've got to explain what's going on, why we get these turbulent states. And then the other thing is that there are lots of different systems that as soon as you put them together, they're actually called active pneumatics, these systems, you get this turbulent sort of behavior. Are there any connections between the different sorts of turbulence? Are they really all doing more or less the same thing? And we sort of hope so, because as a theoretical physicist, I can only sort of manage one thing at once. And so we hope we can unify what's going on in these systems. So let me start by thinking about these guys, the microtubules driven by motor proteins, and just explain to you a little bit more about what you're seeing here. So this is one of my favorite movies. This is inside a cell. Somehow you have to move proteins and other things around a cell. And if you just try and have them swim around the cell, you've had it, because there's a lot of Brownian motion on these length scales. And so they get buffeted all over the place. So the way that the cell copes with this is laying down these microtubules, these long polymers. And these polymers are continually forming and dissolving to form sort of train tracks. Let me play that again while I'm talking, if I can get it to work. Yep. And then the motors walk along the train track, so they're held down so they don't get buffeted around, pulling the proteins in this backpack. So this thing here is a motor protein. This is a computer simulation, but it's pretty much what they do, except that they don't usually do it quite so regularly. It's a bit more sort of forward and backward.
Yes.
Hi, Kevin. Are you? Mute yourself.
I didn't start it. Who's started it?
Oh, never mind.
Kevin, can you mute yourself, please?
Talk to Chris.
Yeah.
I'm listening to the talk.
Oh, so anyway,
Zoom is working as far as I can tell.
Hold on, I'll try to mute him.
I sent the announcement out and I clicked on it and I got Zoom. Bye.
yeah, okay. So what we've got is these molecular motors, and this is a picture of light microscopy of these things, just to show you that it isn't just computer simulations. Here are these little feet, and as a function of time, it's better on this one, you can see them walking along the microtubule. So these are light microscopy. And one of the best experimental systems to look at dense active matter is due to Zvonimir Dodgik's group. And what they did is take the microtubules and end up with a suspension of the microtubules, and then put in double-headed molecular motors, double-headed kinesin motors. And these motors walk along the microtubules, and they walk in a certain direction. They have a directionality to them. And so these guys will walk in this direction and push the microtubule up. These guys will walk in that direction and push the microtubule down. And so the motors are pushing the microtubules relative to each other. And that's what gives this active turbulence. Let me play it again. So here we are. What you're seeing here is bundles of microtubules, these sort of white stringy things, and then they form structures. I mean, it's turbulent, but there's sort of structures in there, right? This is a sort of threefold sort of structure. And here we've got bends, which look a bit like comets moving in this direction. So this is the sort of system that we're going to try and understand. So why bother studying active matter? Well, first of all, this is non-equilibrium statistical physics. People have sort of realized that we can use active matter as a really nice experimental system to look at the behavior of lots of particles, to look at statistical physics out of thermodynamic equilibrium. Another reason is to create new types of tiny engines. These molecular motors walking along microtubules, of course, are engines, and nature is able to build these really rather amazing engines, and we can't do it. We just can't build anything which is that sort of size. And then what I'm going to perhaps concentrate on here is that we're going to try and understand biological systems. Because if you think about it, well, I said biology is out of thermodynamic equilibrium. So are there relations between the way we're looking at this active matter and what's going on in biology? And I think this is a question which is becoming increasingly thought about, but really is very much at the beginning. So what I'm going to do is, first of all, try and explain where this active turbulence comes from. And in particular, I want to introduce the idea of motile topological defects and the fact that any gradients in the pneumatic order leads to stress. So this is a sort of background. How can I describe active turbulence? And then I'm going to use it to describe 2 or 3, depending on time, different problems which we've been thinking about in biology. And I hope I'll convince you that we can see some links between the systems, but we're really very much at the beginning of what we might be able to do with this sort of approach. So let's start with the idea of pneumatic symmetry. So pneumatic symmetry means you have up-down symmetry. You have a preferred direction, but up and down are the same as each other. And if you take a pneumatic molecule or a pneumatic colloid and put lots of them together, what you can end up with is a pneumatic state. And that's a state which has long-range orientational order. So there's this special direction, and on average, everything points along this direction called the director field. But there's no long-range positional order. The centers of these molecules aren't on the lattice, and this is a liquid pneumatic liquid crystal. And I'm going to be using the Q-tensor, which is an order parameter, and you don't have to worry too much about that, just that it's a way of describing this pneumatic order. Its largest eigenvector is the direction of the pneumatogens, and its largest eigenvalue tells you how much order you've got. we sort of know about this at Oxford, this pneumatic order, right? Because in the summer, everything's very disordered, but in the winter, everything, when it's cold, everything ends up in the pneumatic state. So sometimes the pneumatic order goes wrong. And when it goes wrong, you end up with things called topological defects. They're topological defects because the order is going wrong all the way to infinity. You can't do a localized change to get rid of them. And that means they actually on their own have infinite energy. We're going to be in 2 dimensions. And the defects I'm going to talk about most are ones like this red one here, which is a +1/2 defect, which has the shape of a comet. So this thing's going to come up a lot in this talk, and a -1/2 defect, which has this 3-fold symmetry. Now in a passive, in an ordinary liquid crystal, one which is maybe in your laptop screens, these defects slowly move towards each other, and then they annihilate in pairs. And if you're lucky and they don't get stuck as they're moving towards each other, they annihilate to give perfect pneumatic ordering. So don't worry about the details of this, but I'm going to end up solving the equations of motion for one of these active liquid crystals. And we're going to start from the equations of motion of a pneumatic. And really all I care about is you realizing that these are continuum equations. So they're a bit like the Navier-Stokes equations, but they're more complicated because as well as the velocity field, I have this order parameter field. So I have an equation for the order parameter, and I have an equation for the velocity. And this one looks pretty much like the Navier-Stokes equation. Okay, here's the advective term, and here's the gradient of the stress tensor. And these are well known. We don't have to care about the details, but just to know that they're very well known. And the question I'm going to ask is now, if instead of a passive liquid crystal, I have an active liquid crystal, one of these active things, what difference does it make? So let's go back to the swimmer one. This is active turbulence in the swimmers. And picture down here is the vorticity field. You can see you have lots of swirls. That's the vorticity, is the swirliness. Here, the red bits are strong anti-clockwise vorticity, and the blue bits are strong clockwise vorticity. And this vorticity is very characteristic of active turbulence. The sort of length scale here is maybe about 10 swimmers across. So you get these structures, very turbulent-like structures in the flows. So if these were just long, thin things sitting there not doing anything, we'd be able to use those equations that I've just shown you. What extra do we need to make this work for pneumatic systems, for active pneumatic systems? Well, the extra that's happening is that these things are moving, and they're creating fluid flows in the water around them. As they swim, they create flows, and we need to put those flows into the equation somehow to get us from a passive system to an active system. So what do the flows look like around one of these little swimming things? Well, here's my little swimmer, and these are actually experimental results from the other place. And what the swimmer is doing, you can see that the far flow field, it's pumping fluid out from its ends and pulling it in from its sides. And that flow field has this pneumatic symmetry. There's a special axis here, but this direction and that direction are the same. Not close to the swimmer, it wouldn't swim, but in the far flow field, which is what we have to worry about, we have this pneumatic symmetry. The reason for the pneumatic symmetry is that this is an active particle. It's a self-propelled particle, and so all forces have to be in equal and opposite pairs. And the result of forces being equal and opposite pairs is that this ends up giving you a far flow field which has nematic symmetry. So how do we take this equation and make it active? And it turns out this is actually very easy. You just have to add this one term here, this active stress. The active stress is equal to zeta. This is just a coefficient saying, how many swimmers have I got? How strong is this activity? And then this Q, this order parameter tensor for a nematic field. I certainly haven't proved why that's Q, but it seems reasonable on grounds of symmetry that that would be the leading order term in some sort of continuum equations because of the symmetry that these particles produce, the symmetry of their flow field. And the important thing about this active stress, is where it comes in the equation. The active stress comes in here in this stress tensor, and it comes under a derivative. So that means the minute there are gradients in Q, gradients in the nematic order, you're going to get stresses and you're going to get flows. So in words, this is any gradients in the magnitude or the direction of the nematic ordering gives you flows. So what? Well, the most important thing is that these active flows destroy the nematic order. And this picture will show why that's the case. Here I've got my swimmers all nicely lined up in a nice nematic state. Let's say there's a tiny fluctuation, a tiny bend fluctuation, which gives me gradients in the direction of the nematic order. Gradients in the nematic order give you stresses, give you flows. And if you work it out, the flows are in this direction. And so what's going to happen is that bend, these changes in the nematic order are going to increase. And what's going to happen is the whole thing is going to be destroyed. The nematic order is going to be destroyed due to the active flows. In maths language, it's if you do a linear stability analysis, the nematic state is unstable to activity. There's a very nice experiment showing that from the Barcelona group. So the problem with real systems in actually showing this instability is it's hard to get them lined up in the first place. So you have to get them lined up before you can see the instability which starts the active turbulence. And the clever idea in the Barcelona group was that they had a straw and they sucked through the straw, and this is the microtubule motor mixture, and the microtubules all lined up pointing to the straw. So some poor graduate student was sort of sucking this stuff up, right? And what you'll see is that everything is lined up pointing towards the straw, and then as soon as the graduate student stops sucking, the instability starts. So I'll play it twice because it's pretty quick. All right, so this is everything getting lined up. And there you can see the instability starting. Let me play that again. Again, let's say this is the microtubule motor mixture. First bit just shows everything lining up. And then very quickly, this instability starts. And if you look carefully, you can actually see a secondary instability where you get bends perpendicular to the first ones. And gradually the fluctuations mix everything up and you end up with proper active turbulence. And if we solve the equations, this is what we get, this vorticity field, exactly like the vorticity field that I showed you before. And if we sort of home in so we can actually see each individual little swimmer, what you can see here is that each of these are the swimmers. You get these bends in the order parameter, bends in the nematic order, and that's what's driving the flows. That's what's driving this messy, turbulent flow. So I've been on about this being a pneumatic. And I said that pneumatics had topological defects. So what happens to the topological defects? Let's run these simulations and put a red dot every time I have a +1/2 defect, that was the comet-like ones, and a -1/2 every time I have these threefold defects. This is this one here. Okay, so let's run the simulations and see what happens. And what you can see, if I get the right ones, is that these defects, which I didn't do it, they're meant to annihilate in plus and minus pairs. That's good. These guys do too. But then if you look up here, you'll see something new happening. That as well as annihilating in pairs, you get them produced in pairs. That wouldn't happen in a system without activity. That happens because of the activity. You get these topological defects popping out. It makes sense, that's fine, because the whole idea of an active system is that one's pumping in energy. And so this gives you the energy to overcome the elastic energy, which is trying to line up the liquid crystal to produce the defects. If the defects were produced in these pairs and just sat there, it wouldn't work because they'd annihilate each other again. But once the defects are produced, just try and play it again, oops, going backwards, they move away from each other. So you can see that they're produced and then they move away from each other. So the defects, as well as being moved around by the background, are also self-motile. They move on their own. And actually that makes sense, right? Because I've said that any gradients in the order parameter give you stresses and flows. And they're very definite gradients in the order parameter here. And that means that this thing is going to produce a flow field. And because it isn't symmetric, it's got a preferred direction. So it will set off in one direction or the other. This one also produces a flow field, but because it's symmetric, the flow field won't make it move anywhere. The next slide, I've got a picture of those flow fields. Here we are. This one's a +1/2 and it moves here in this direction, and the flow around it forms two vortices. This one is the -1/2 and it sort of just sits there, but produces flows, which are these 6 vortices around it. And we'll see that in a real system in the first example I'm going to talk about. This is back to this microtubule motor protein system to actually see the defects in a real experiment. Here, what's happening is that the active forces are bending the microtubules, and this is equivalent to creating here a +1/2 defect with this comet-like shape, and a -1/2 defect here with this threefold symmetry. And you can see that the +1/2 is moving, and the -1/2 is just staying put. This is really what we've seen lots of times, but with the defects actually marked in green. The +1/2 defects are marked in green. More pictures of the turbulence. These guys are just better fed. There's more ATP in this suspension, and so you end up with more defects and a smaller length scale for the vortices. Okay, so that's the introduction. This is active pneumatics, and the punchlines are that in these active pneumatics, any gradients in the order parameter give you stresses and give you flows. And we get active topological defects, and in particular, the +1/2 defects are self-propelled. I just wonder if anybody's got any questions, I'd be happy to answer them now or later. All right. Okay, so now let's look at 3 different examples of how this idea of active pneumatics might impinge on what's happening in a biological system. And the first one is the story of the hare and the tortoise. And thanks to the people who contributed to this, in particular, Ollie did the experiments and Armin was the student, postdoc, who did the theory. And this is another active system. This is Pseudomonas, which is a sort of bacterium, and they look very pneumatic-like. They're sort of like rods. And what they do is they twitch. They move around on the surface, pulled by pili. So they're not actually swimming things, but they're twitching things. And they sort of move forward and backwards on the surface. And you can definitely see here the short-range pneumatic order in these systems. We got into this because my zoology colleagues were interested in what happened when you had a mixture of two different sorts of bacteria, and in particular, one which was the wild type, which I eventually managed to realize meant straight from the pond rather than fierce, and one which was genetically modified to have extra pili. So this one's got more little twitchy things, and so it goes faster. So we had a slow strain and a fast strain, and we wanted to know how they spread out. When you mix them together, how do they spread out? So this is just seeing them spread. There's great colonies of them off to the right-hand side of the screen. And so we're going to see how they spread. They don't divide very much. So this is really just motility, which is making them move. The top ones are the slow ones. These guys are the fast ones, and these are just a control where the pili have been removed. So you need to look at these two up here. And what you can see is that top ones are the slow ones, and these are individually the faster ones. So the slow ones are the ones, whoops, I want to go backwards, so I can play that again. The slow ones start slower, but eventually overtake the fast ones. This is the hare and the tortoise story. If we mix them and watch them expand, you see the same sort of thing. Down here, this is what happens as a function of time, looking at the front of a colony, which is mixed. The fast ones are yellow, and they dominate the colony edge at early times. But later on, the slow ones, which are blue, are pretty much all there is at the edge of this colony. So what on earth's going on? Why are individually slower bacteria actually ending up moving faster? I mean, by here, you can't even see the fast ones. So let's just think about defects in this system. It's a pneumatic, it's an active pneumatic, so I ought to be able to identify defects, so we did that. The red ones are the +1/2 defects, and the blue ones are the -1/2 defects. Looks sensible. This is the experiment. We could do simulations to see this rather easily, because the bacteria are behaving like hard rods. And as they move around, I mean, you can simulate this very easily. You can just have hard rods which can't overlap each other, and each rod is moved by a constant driving force. Again, you can see very much the same patterns. You can see these + or -1/2 defects. And I played this before and I'll play it again. This is the continuum model of the same thing, + and -1/2 defects. So if I were you, I'd be saying, fine. Okay, so they're defects, but why are they, the same sort of defects? Why are these very different systems anything like each other? So sort of to check that, we looked at the flow field around the defects. We looked at the flow field around a -1/2 defect on this top line and around a +1/2 defect on the bottom line. And we compared the continuum simulations, which I showed you before, with the rod model, the rod simulations, and with the experiments. And what you can see is pretty much the same thing. I mean, these are much noisier, and those are even more noisy. But you see these six-fold, this six-fold symmetry of the flow field. 1, 2, 3, 4, 5, 6 there, and 1, 2, 3, 4, 5, 6 there. And here for the plusar halves, you see two vortices here and here and here. So it leads to a good approximation, at least a sort of first approximation. These things are behaving in the same way. So what's the link? What's the link between this hare and tortoise story and topological defects? Well, when we looked at the bulk of the colony, what happens is that as a function of time, you get these blobs forming. And now we're starting from the beginning with different experiments. And again, you can see these very solid blobs. And what these blobs are is regions of fast bacteria, predominantly fast bacteria, that are stuck. They're stuck with the bacteria pointing outwards from the surface. They've been verticalized, sorry about the word, but they're pointing outwards to the surface, so they can't move anywhere because they just get stuck in mounds. And I'll tell you the reason they get stuck in mounds and then show you the evidence. The reason they get stuck in mounds is that 2 plus a half topological defects come towards each other. That makes a virtual +1 defect. That sets up flows, and the flow field it sets up push the bacteria out of the plane. Here's a side-on view. These defects come together and they push the bacteria out of the plane. The bacteria get stuck, and then other bacteria sort of hit into them and get stuck and form these mounds. So why does that work better for the fast bacteria? Why does this get rid of the fast bacteria and not the slow ones? And the answer is that if these defects are moving fast enough, they can do this, and fast bacteria have fast defects. If they're moving slowly, they sort of push things up, but there isn't enough energy to keep them standing up. This is a simulation model, which, if you like, is a bit of a cheat because you can get the parameters to do the right thing, but it's a good way of showing you an example of what happens. The one on the left is slow, the one on the right is fast. The slow ones get pushed up and fall back down. The fast ones keep pointing towards us out of the plane, and then they get stuck. They can't move anymore. And we've got a bigger simulation here showing how with fast bacteria, things form these stuck regions of verticalized bacteria. With a slow bacteria, it doesn't work. So this is the real thing. This is the real experiments. These are amazing experiments from Ollie, because actually finding the right place to look for a couple of these defects coming together is tricky. I'll show you the movie. This is, yep, that's it. Here's one of these, we call them rosettes being formed, one of the mounds being formed. And it's probably easier to see with the color in. Here's the mound. Here's a sideways view of the mound. And the color coding is that the fast bacteria are yellow, and you can really see that it's the fast bacteria that are forming in these mounds. So that's the conclusions. I'm not in any way saying that topological defects are responsible for the whole of biology, but for this particular thing, it seems that things have evolved not to move too fast, because if they move too fast, the topological defects interact and push them away from the surface. Okay, so that's our favorite movie. I do want to show you just a couple of other examples before we move on to the next topic where we see topological defects. This is Hydra, which is a marine polyp, maybe about millimeter high. And Hydra is a wonderful thing because you can cut out a bit of Hydra about this big, a really tiny bit, and it grows back into a fully sized Hydra. And there's some very beautiful work by the group in the Weizmann who show that the arms grow at positions of topological defects, and also that the mouth up here and the foot also grow at positions of topological defects. So we tried to solve the active pneumatic equations of motion to see what would happen for a shell, an active shell. And what we find is, indeed, we get arms forming, not so regular ones as Hydra, but you get these arms forming. And the reason they form is that they form at positions of +1/2 defects, because +1/2 defects move, they create flows, and so they push the shell outwards. And then when they escape from the ends of the arms fall back in again. I think we'll move on there. And so starting from there, I wanted to think a little bit about active changes in shape. So up to now, I've been talking about 2 dimensions. And what we want to do now is think about 3 dimensions and say, if I've got a ball of active material, it's being active, it's producing all these flows, what's it going to do? So let me start again. Oh, I should say thank you to Liam, who spent lockdown doing this work, absolutely brilliantly, completely self-motivated. And also some beautiful experiments now. It was only 2 years ago that people were able to make an active system in 3 dimensions, a fabricated active system in 3 dimensions. And this again is this microtubule motor protein system, again from the Dogic group. And what they had to do is take the active microtubules and add pneumatic colloids, because the stuff needed to be helped to form a pneumatic phase. And if they did that, they found active turbulence, and in particular, the difference in 3 dimensions, it's a movie, the difference in 3 dimensions is that instead of just point defects, you get loops, you get dislocation loops. These loops are topologically protected again. They can shrink and disappear and they can grow, but they can't sort of split up. So they have to end either as a loop or end on the edges of the system. And the way I like to think about it is that the cross-section of these loops are a bit like these point defects in 2 dimensions. So here we are. Here's a blob of active material. We're back to simulations now. It's a blob, it's a droplet, if you like, of active material. And we can see the disclination lines which are formed and move around, and they're pushing the drop, but here we've got quite a large surface tension so that they don't push the drop too far. So I now need to go back. Remember when we looked at active stress, the active stress was zeta Q, and zeta can take 2 signs. if zeta is positive, one has an extensile system. It corresponds to a pair of forces pushing outwards. And if zeta is negative, you have a contractile system, which corresponds to forces pulling inwards. And if you have an active drop, you have something called active anchoring. And that means that the swimmers or the pneumatogens or the microtubules line up in a certain way on the surface of the drop. There's no thermodynamic anchoring here, no free energy or anything. Just the activity will give you anchoring on the drop surface. And the reason is, again, exactly the same thing as we said before. The reason is that inside the drop, you have a pneumatic, and outside the drop, you don't. And so you have a change in the magnitude of the pneumatic order parameter over the edge of the drop. And a change in the order parameter sets up stresses, sets up flows. And it turns out that the flows tend to push the pneumatic to line up in plane, parallel to the surface if you have zeta positive, and normal to the surface if you have zeta negative. And that sort of makes a difference when you look at the behavior of these drops. What I'm going to show you is that, and I'm going to really just do it with movies, if you have a contractile drop, you end up with something which looks like a walnut. You get lines which sort of go up and down. If you have a contractile drop which is rather small, you end up with invagination. And if you have an extensile drop, you get these arms forming. So let's start with the extensile drop and look at the formation of the finger-like protrusions. This is more or less what we saw before. These are caused because defect lines, which look a bit like plus or half defects, push bits of the drop outwards. And indeed, that's actually been seen in experiments a long time ago. This again is an active shell. And if you wait a bit, you'll see arms coming out. And those arms are formed at the positions of plus or half defects. It's plus or half defects trying to escape. Their arms are much better than the ones in the simulation, and that's because it's hard to get a big enough drop relative to the arms in the simulations. This is now what happens in a contractile drop. Here, the anchoring is contractile is normal to the surface. And you can see this thing forms a walnut-like shape. And again, it's because of the places where the topological defects hit the surface. They're trying to minimize, they're trying to agree with the active anchoring. And they do that best by joining up in pairs and then pushing the surface up or down according to those pairs. The one I like best is this one. This is a tiny contractile drop. So this is a drop which has normal anchoring on the surface. And you can imagine it's really unhappy because in the center of this drop, you're going to have +1 defect, in fact, but a place where the elastic distortions are enormous. So this is a very unhappy configuration. So as soon as you turn on the activity, what happens is that you get a band around the surface where the surface anchoring becomes planar instead of normal. This sets up forces, this sets up flows, and what the flows do is take this drop and cause it to invaginate. So the drop moves so that it ends up in a cup-like shape. This is done just by activity. And this, of course, looks very like what's happening in the early embryo. Embryos start with a ring of eggs with a space in the middle. And one of the very early things that happens in embryogenesis is that this thing invaginates to give several layers, usually 3 layers, which are going to differentiate into different sorts of cells and grow into different parts of the body. And so this early gastrula is looking very like what we're seeing in the active system, although of course real biology is much more complicated. And one of the things we're trying to do at the moment is make closer links between what's happening, us and many other people, make closer links between the ideas of active matter and what's happening in these systems. So my last example is one which we haven't published, which we're doing at the moment, and where I'm still thinking about what the right thing to do is, and that's cell sorting. Because if you have, that's the disclaimer. So as to shut up any biologists in the audience before they start telling me what's wrong with this. And thanks very much to Saraswat, who's the person behind this work. And in particular, the simulations are complicated, and he was the one who worked out how we should do it. Okay, so this is the story. If you have two different sorts of cells, and now I'm talking about epithelial cells, cells which are plated on a substrate, the sort of cells that line the body, but here they've been taken out and put and been plated on the substrate. And this is a mixture of epithelial cells, which are the wild type cells, the normal ones, and ones which have had the cell-cell junctions changed, so they work in a slightly different way. And if you put down a mixture, what happens is that you get phase separation into two different cell types. And I wanted to understand whether this could be driven by activity. I mean, we're all used to the idea that if the interactions are different, as in a mixture of liquids, you can get phase separation into two different liquids. But could this just be due to activity? And so what we did is that we looked at a two-fluid model where we had a mixture of an isotropic liquid and an active pneumatic. We made sure that the free energy wanted them to mix. So we had a single well free energy so that there was absolutely no thermodynamic reason for them to phase separate. And then we coupled the two fluids by a friction term proportional to the difference in velocities. That's obviously a physical way to do. If you have two things moving at different velocities, the friction will bring them back to the same velocity. And we said, can this active system give me, so it's driven by this active pneumatic, can that give me phase ordering? And this is the answer. Here's the movie showing how this thing orders into a pneumatic rich phase, which is yellow, and a pneumatic poor phase, which is blue, and everything's moving around because you've got active turbulence underlying what's going on in this system. So why? Can we explain what's going on here? And it's the same story again. It's a mixture of flows being set up at concentration gradients and active anchoring. Here's how it works for an extensile system. For an extensile system, the pneumatogens line up parallel to the surface. Here's my surface between my active region and my passive region. So the pneumatogens line up parallel to the surface. That's really because of the flows they set off in this direction. That's active anchoring. These are extensile things. Extensile things have flows outwards from their long ends and inwards from their short ends. So if I put this on here, inwards from the short ends, I'll put them on, we get flows in this direction. And on this side, you get flows in this direction. But because there's a concentration gradient, there's more flow here than here. And so I'll get a net flow up from the passive side to the active side, which will drive the pneumatic into this region here and is responsible for the phase ordering. You can see here that the degree of phase ordering is plotted as a function of the activity divided by the friction between the fluids. And this makes sense because if you make it more active, you're going to get more phase ordering. And if you increase the friction between the fluids, they're not going to have so much of a relative velocity. And so you're going to get less phase ordering. And what we want to do next is use this two-fluid model to see if we can look at more complicated systems. And so let me finish by showing you a lovely set of experiments, which is nothing to do with us, but I think are fun things to try and model. This is the starfish embryo. And the starfish embryo organizes itself into two different sorts of cells, green and red. So this is a nice organized starfish embryo down here. What the experimentalists did is just mix this up so that the green and red cells were completely mixed up. And then they waited to see what would happen. What we see is phase ordering. The red cells tend to cling to the red ones, the green ones to the green ones. You can see these phase ordering happening here. And then something which wouldn't happen, I think, in a passive system, things get even more complicated and you get this extra structure in this system. And so that's probably trying to understand this, and also mixtures of two different active fluids is one of the next steps. I've talked about active turbulence. In nature, that probably isn't the right story because we don't want things to be turbulent, we want them to form patterns. So I think, again, a nice new direction. This is Drosophila, which people have looked at, the fruit fly embryo. This is a movie over several hours of the fruit fly embryo growing into a fruit fly. And what you can see is flows, and you can see pattern formation. And I'm sure there's lots of biology in there, but I think this shows that there's going to be quite a lot of physics in there as well. And it also shows that although we've seen some simple examples, we've got a long way to go to explain what's happening in things like this. So thank you very much for listening.
Thank you for a very interesting colloquium. Let's see, we should allow people to ask questions. I think the simpler thing is for anybody who wants to ask a question, just speak up, and we'll just go in the order of time. So the first one to speak up gets to ask the question. Well, that didn't work. How about raising hands?
Sumi, I can see the hands there. Yeah.
Hi, yeah, thank you. That was such a great talk. I really enjoyed it. I have a question though from way back when, and that is, you have something that's linear in Q that is active stress. And is there an easy way for someone to understand why that works? Because it seems, at least, you know, in any of these movies that you're showing or any of the simulations, there's just the flow fields must non-linearly interact somehow, because the swimmers or you know, the microtubules are so close to each other that I would imagine you would need some sort of higher order active stress term.
I mean, if you think of each of these things as an extensile swimmer, so you look at the little forces and you work out the leading term from that, and you coarse grain, so you're averaging, then that is the leading order term in the expansion.
And so
it
when
seems to be enough. I mean, if you add a Q squared term, which you could have in there, it's, you know, symmetry allows it, it doesn't make a lot of difference.
okay, so how big is your coarse graining? How so how many swimmers across, or how many things across?
I mean, it seems to work for not very many swimmers. I mean, obviously you need it dense. So in a way, because it's dense, you're looking at the flow field due to several swimmers, due to a lot of swimmers at once. So it's more denseness than size, I think. You lose the active turbulence when you confine them too small, and that's just because you kill the instability.
Could you show us those equations again?
Yes, a long way though.
Yeah. Oh, wow. Boy, there was a lot of slides.
Nearly there. Yep.
Yeah, there they are. Okay. So the top one is linear in everything, and
Well, no, because
but there's a U there that multiplies the Q. What is U again?
use the velocity.
Oh,
So
okay.
this is the advective term.
All right, so that's so the top one's nonlinear also, and the middle one is nonlinear,
Yep.
obviously.
So this S is the way that the pneumatics turn in a shear, and this is a relaxation term which puts you down to a thermodynamic minimum of a free energy. But you never get there because this is the active system. And here you've got all sorts of very strange elastic terms in the stress tensor, which come from it being a liquid crystal.
Yeah, and I think that's what was, so the last thing, the last red equation is what I was stuck on, is why does it work with an active stress that's just linear in the order parameter tensor? But I think what you're saying is it obviously works, but it's probably because of you're averaging over like a macroscopic flow, not individual swimmers.
Yes, that's right. I mean, to get this, you're doing a coarse graining. But if you are going grounds of symmetry, that's going to be the leading term.
Thank you. You can just put it there. Thanks a lot. You too.
So do you actually do the experiments yourself, or you various people in your group do experiments and others do the theory or the modeling or the
No, I mean, we're theorists. I mean, I hope I put the names on. Most of these experiments were either Dodgik's group, who's now at Santa Barbara, or the Barcelona group with Francesc Sagres. So the theory is ours, and indeed with the background theory, many people's.
So you do the theory, but you are communicating closely with the experimentalists, or you just read their papers and interpret them?
Well, both really. I mean, so the tortoise thing is a collaboration with experimentalists, and we've done quite a lot on with the Barcelona group as well. So then, I mean, not all of these papers, but a lot of them are done jointly.
Well, let's see. It's gotten to be 11:30, and in England, if there's no further question, I think we ought to let Professor Yeomans get some rest. It's very late over there. So I'll end the thing here then. And we all are very, I've seen various comments show up that people are delighted with this talk, and it's given many of us a whole new perspective and way of looking at things. And and perhaps helped us to look at life differently. So once again, thank you, and I hope you get some well-deserved rest.
Okay, thank you very much. Thanks for listening, everyone. Okay.
Thank you.
good night.
Bye-bye. Thank you.
Thank you.